Hello, everybody. Welcome back. It's 12:20. So I think in the interest of time, we should probably get started with our next set of sessions. We had a really wonderful set of talks this morning, and this will continue with our next set of talks. So without any further interruption, so I'm going to introduce our next speakers. So this will be a tandem talk given by Dr. Liz Buttermore and Dr. Nadif Kasri. Dr. Buttermore's talk will be recorded and then it will be followed by Dr. Kasri's talk, Harnessing the Potential for Stem Cells to Understand Kleefstra Syndrome, as well as the next talk right after that, The Promise of iPSCs, Patient-Driven Research for Rare Neurodevelopmental Disorders.
Disorders. So there are some challenges facing drug discovery that are unique to the area of neuroscience. These include the ability to access the brain through the blood-brain barrier, and also that the biology of the central nervous system is very complex, and we're actually still just understanding the circuitry, and it's still just being defined. There are a few validated targets for neuroscience indications, and many of these are decades old or ineffective. And because of these challenges, large pharma has largely been taking a step back from neuroscience research. But that allows us the opportunity to fill in the gap in this preclinical space. So how can we get better clinical results? It starts with better preclinical data. And that starts with the model systems that are being used to generate the data. So traditionally, rodent models have been used in neuroscience, and you can see that the mouse brain is very different than the human brain in complexity. In addition, rodents don't contain the genetic diversity that humans have. Other cell models that have been used in neuroscience preclinical research include engineered cell lines where particular targets would be overexpressed and screened against. But these cells don't recapitulate the fundamental functioning and property that are seen in neurons. We really need to be using neurons to model neurological disorders. So fortunately, we live in the time of stem cells. Stem cells are undifferentiated cells that have the ability to become different types of cells, and they have the ability to self-renew indefinitely. There are several different types of stem cells, but today I'm going to focus on induced pluripotent stem cells, or iPSCs. These are pluripotent. They're made, which means they have the ability to become any other type of cell. They are made from skin or blood, so they don't contain the ethical considerations from using embryonic stem cells, and they're patient specific. So we have the patient mutations and genetic background from patients in order to study their cells in a dish. So what can we do with these stem cells now that we have them? We can differentiate them into different subtypes of neurons. And what this allows us to do is to model the type of neuron that might be affected, how we can start from stem cells and make different types of neurons in a dish. There are some strengths and limitations to using this technology. The strengths include that they're human, they're scalable. As I said, we have patient-relevant genetic backgrounds. The neurons that are made can be maintained in vitro for months, and some have shown even years. They're genetically modifiable, so we can correct patient mutations. And we can make some well-defined cells via differentiation. We, in the field, have also been moving towards the use of 3D organoids, which will allow for further maturation and studying these neurons in a space that's even more similar to the human brain. For limitations, the neurons we make are very embryonic and immature. They're prone to karyotype or genetic changes. There's a lot of variability in this technology, and we're focusing a lot of efforts on trying to understand the sources of this variability and how we can control it. It's relatively expensive and labor intensive, and the exact identity of the cell types that we're making are not precisely analogous to those in patients. So while we're making cortical-like neurons, they're not going to be exactly like the cortical neurons found in a human brain. With these strengths and limitations in mind, we have established this platform where we are collecting patient fibroblasts or blood samples, having these reprogrammed into patient-specific iPSCs. We also either then genetically correct a patient-specific mutation or also collect a control line from a familial family member. We then differentiate these iPSCs into different subtypes of neurons and try to understand the phenotypes in the patient-derived neurons that are different from healthy control neurons, either in morphology or in the function. Once we've identified these differences, we can then use them to develop screens and either use them for drug repurposing screens or novel target identification screens to understand the biology of the disease better. These compounds are then able to be tested for efficacy and safety in animal models with the goal of returning back to the patient in clinical trials. I made a note here also to remember to mention that, as you'll hear today, it's really important for us to also have really strong natural history information and biomarkers so that we have the best chance for success in the clinical trials. And this platform has already been successful in other areas of neuroscience. So this is one example from the field of ALS, where iPSCs from familial ALS patients were differentiated into motor neurons. They were shown to be hyperexcitable, so they fired more in a dish, and this led to increased cell death in the ALS motor neurons. This group of researchers ran a screen and identified retigabine, which is an FDA-approved drug for epilepsy, and it found that it decreased the hyperexcitability and increased survival of the iPSC-derived motor neurons in a dish. And this has now moved into phase 2 clinical trials based on this work. So this is really exciting for us, and we are now have the goal to do the same thing and fill the gap for rare neurodevelopmental disorders. So far, we have collected blood and fibroblasts from over 540 patients and controls, and we have iPSCs from 119 patients and controls. These patient lines have been collected from Kleefstra syndrome, PSC disorder, SSAGH deficiency, CDKL5 disorder, and other epilepsies, early onset psychoses, autism-related disorders, and developmental disorders. And today, I just wanted to take the 2 minutes that I have left to tell you about how we're using these patient lines from TSC to complete the story from patients to the cells and back. So TSC, or tuberous sclerosis complex, is caused by mutations in TSC1 or TSC2 genes. This can cause tumors in many parts of the body, including the brain. And there are various different symptoms that can present in these patients and include things such as seizures and cognitive disabilities, including autism or ADHD. And we wanted to see if we could model the seizures that are seen in these patients in a dish using the iPSC-derived neurons. And the seizures are thought in part to be caused by imbalances in the excitation and inhibition in the brain. And so we first wanted to see if we could detect these differences in excitability. So here we have 3 iPSC-derived neuron lines. First, the healthy control. The second is a patient with TSC2 mutation, and the third is a homozygous deletion of TSC2. And what we find is that loss of TSC2 results in increased hyperexcitability or increased firing in these iPSC-derived neurons. So here you can see in the red line at the top, the increased firing in the TSC2 mutant line compared to the blue line at the bottom. And what was exciting is that when we treat with rapamycin, we're able to prevent this hyperexcitability in these iPSC-derived neurons. And this is really interesting because rapamycin, an mTOR inhibitor, has also been shown in rodent models of TSC2, where they can place an EEG cap to record seizures from rodents, that in the gray bar here, you can see treatment with rapamycin reduces seizures, whereas vehicle treated in the black bar, they continue to increase their seizure frequency over time. Rapamycin has also been shown in patients to reduce seizure frequency, as you can see here from this clinical trial. So what this tells us is that we now have a model system for TSC2 where we can screen for other compounds to reduce this hyperexcitability and see if we can identify things that might reduce seizures in patients, but maybe not have some of the side effects that current treatment options have. What I hope I've shown you today is how we are able to take patient cells, reprogram them into iPSCs, differentiate them into neurons, and then use these neurons to model the disease in a dish to better understand molecular mechanisms of the disease, and then also to run screens to identify new drugs that can then come back to the patient. And what you'll hear about in the next talk from Neil is how his lab has done some really great work to start modeling this disease and understand Kleefstra syndrome on a level that we haven't previously been able to see. So with that, I want to thank the leadership of the core, Mustafa, Clifford, and Mira, other members that keep our core running, and of course our funding. And thank you all for your attention. Hi, I'm Liz Buttermore from the Human Neuron Core at Boston Children's Hospital.
Great. And so we can save the question and answers for the chat box. Let's have Dr. Nadif present.
Yeah, I hope you can hear me. Can you all see my screen?
Hear you and see the screen. Yep.
Okay, perfect. So it's really a pleasure to, well, first of all, to be able to show part of our work on Kleefstra syndrome today, and also to follow up on Dr. Liz Buttermore's work. She has done a very nice introduction, and would like to continue on this actually, and how we try to harness the potential of using these human cells in a dish to study or to model Kleefstra syndrome. And so this is really work that we've done also at the Human Genetics Department in Nijmegen, together with Professor Kleefstra and Professor Hans van Bokhoven. And as mentioned by Dr. Buttermore, the ability those days to collect blood samples of fibroblasts from patients and to convert those back to stem cells really allow us now from in the dish to generate the cells we are really interested in, and those are the brain cells. And this is really unprecedented because now we can follow the development of all types of brain cells in a dish in a laboratory and study those and manipulate those in a very safe way without affecting the patient themselves. And so I would like to show you today or share some of the data or some of our work of the past few years and how we have done this. And so, one of the most important things, of course, is to be able to generate those brain cells. And there is a lot of work being done on this research, and it's still improving every day. And as it was mentioned before, those brain cells are still relatively immature compared to the real brain cells, but yet they still get really some very nice properties. And this is one of my favorite images, and just showing you how we can actually generate those what I think beautiful brain cells. As you can see here in green, you can see all the what we called the dendrites, or the protrusions from those brain cells, and you can see they're very complex. And in red, you see these little dots on those dendrites, and this is actually where neuronal cells communicate with each other. So they need to communicate with each other, and they do that via those little protrusions here that you see in red. And so this really shows that we can generate cells, brain cells in a dish, and this from every individual patient. And so what we actually want to do is be able to compare those brain cells that we have in the dish. So we would like to compare those from Kleefstra syndrome patients and compare them with what we think healthy controls. Now, of course, the real advantage of doing this type of experiments is that we know that the brain cells that we have in the dish have exactly the same genetic background of genetic information as the donor from which we got the original fibroblasts or blood cells. And this is, of course, really important, because we know there is a lot of variation, there is a lot of individual variation, and everybody is different genetically. And of course, we also know that Kleefstra syndrome patients do have specific mutations in this EHMP1 gene. Now, the program that we have made is really to look at how those neuronal cells can communicate with each other. So, because that's the essence of what brain cells do. They communicate with each other, and that's how they actually carry over information. And so what we have learned from many years of research, and this is from fly work and from rodent work and also from human work, is that very often in these developmental disorders, the communication goes awry. So there is something going wrong with the communication. So in order to measure the communication between neurons, what we did is we actually grow those neurons, not in a normal petri dish, but in what we like to call a smart petri dish. Now, those petri dishes, they look like this. So they are just glass petri dishes, but on the bottom they have electrodes, and those electrodes they allow us to measure the communication, the electric activity that arises when brain cells communicate, to pick them up and to measure this. And in reality, it looks like this. So you can see here on the left a very and zoom in picture of those petri dishes with the electrodes, the black spots, and you can see probably this kind of weird shapes. Well, those are actually the human cells that are growing on this electrode. And you can see me here holding one of those electrodes, so you can have an idea of the size of this. And the nice thing is that when we do this, after a few weeks in culture, we can really literally pick up the activity, and you can see here this all kind of colors happening here, and the white dots are actually the electrodes, and you can see the activity arising. And this really allows us to now measure this activity and how those brain cells communicate. And of course, this is a quite complex endeavor to analyze all this data. But essentially what we do, and I think this is really important, is that we can monitor when brain cells communicate, how often they do so, with who they communicate, because they have always a preference with which cells they communicate, and also how loud they communicate. And this is really a great tool that allows to do this not at one single time point, but also during development. So we can really follow the same cells over and over again and see how the development might be impaired. So now you might wonder, how does the communication then looks different between control, healthy control, and Kleefstra cells? So that's something that we did over the past years. And so the first thing that I think was very interesting for us and really important is that we first found that if we look at, and we call this a raster plot, and don't worry too much, but basically this raster plot tells us how the communication happens. And we looked at how this happens within healthy controls. And you can see here a lot of vertical black lines. And this basically means that these neurons are communicating exactly at the same time. And I think you can appreciate that this happens in a very regular fashion. And also, I think you can appreciate that there is very little difference between the three example plots that I show you here. And those are three completely independent healthy controls. So the real question was, how does it look like when we take Kleefstra patient neurons? And so here we see a comparison on the right. We have three what we call activity raster plots from Kleefstra patients compared to controls, healthy patients. And I think you can readily see that these patterns that you see here on the right are quite different than the patterns you here see on the left. And so what we can do is we can extract the neurophysiological changes or parameters that are different between controls and Kleefstra patients. And so we're in a way, very pleased or excited to see those data because first of all, it tells us that there is a difference. There's really a difference between Kleefstra syndrome patients and controls. So that means that we can measure something different. We have a phenotype that we can measure now in a dish. And the second reason why we were excited is that these differences seem to be very similar amongst the Kleefstra patients. So we see here 3 different patients, and those patients are completely, they are not related to each other, yet they show this very typical same phenotype. And this indicates to us at least that there is what we call the signature for Kleefstra syndrome patients. And this, of course, gives a really nice entry point in trying to understand what is really underlying this. And that's also exactly what we do in the lab. So we try to understand what is causing this different pattern compared to controls. And if we understand this, we can, of course, also try to revert this and rescue this pattern. And that's something that we have done over the years, and we are still doing with quite some people in the lab. And this is just one example where we have actually been able, and this is very recent work actually, where we've been able to take cells from a Kleefstra patient and actually use, based on our biological work, we use certain antagonists for a certain receptor, and we're able to rescue the phenotype that we saw in the dish. And that's something, of course, that we find very hopeful, because it basically is a proof of principle that we can use this strategy now to find compounds that might be beneficial for the patients in the end. And I think one important thing to mention is that all those experiments at the end are done in a dish. We started with cells from, of course, one of the patients, but after that, all those experiments are done in the dish, and they are relatively fast. They can be scaled up, as Dr. Buttermore just explained, and ultimately can now be assessed in a high throughput manner. Further, they are completely safe for the patient, so we don't have to try a drug or compound on a patient. We can try them on the cells without affecting the patient themselves. And of course, they're also patient specific. Now, you might wonder, for the last few minutes that I have left, I also want to share some data that are not from Kleefstra syndrome patients, but are to related syndromes. As we have heard today, there are many rare disorders. And so one of the questions we had quite early is we wondered whether the specific pattern of altered communication that we see in Kleefstra patient, whether this was seen in also other developmental disorders, or whether it's very specific for Kleefstra syndrome. And so we did very similar experiments. And I just want you to look mostly here on the left first, where you see again all those different patterns, where on top you see the healthy pattern, and then beneath that you see all different disorders, the rare neurodevelopmental disorders, Kleefstra syndrome. We already heard about Coulon-de Vries syndrome and also Kabuki syndrome. And I think you can appreciate that none of those patterns look alike, so they all seem to be different. And this is also what we observed is that, and that it is shown in those colored dots here, where every patient line is actually a color. You see that the colors group together. So that means that we have a very syndrome-specific pattern, but that the patterns differ from each other. But there are also patterns that look very similar. For example, Kleefstra syndrome patterns look quite similar to Kabuki syndrome pattern. They are not similar, but they are very close. And this also gives us, again, another avenue to look into the molecular mechanisms of those different disorders, but also learn from each of those disorders. Now, So in short, I think what does it really mean? Well, I think that those stem cell based experiments really allow for the first time to look at to be able to study individual neurons, so neurons from individuals. And I think this is relevant because we've seen already in the beginning of today that there is actually still quite some variation between the individuals with Kleefstra syndrome. And so being able to look at each other's brain cells in the dish allows us also to detect potential mechanism behind this individual phenotyping. In a second step, I think where it will be very important is that whether there will be potential medication, we will be able to test this at the individual level. And we will hopefully be able to use this really as a predictive validity. So be able to see whether a potential compound that works for one person might also work for the other person. And then to finalize, I think that what is important also to remember, and I think that is what makes this day so great, is that we see all different facets of research. And I think we have to realize that each of these research cannot exist without each other's research, in the sense that also this stem cell research really, everything we find, we want to validate that also in a more complex systems, such as a mouse or a fly, where we can also see whether, if we identify a certain compound that can indeed work in a more complex, of course, whole brain organ, and not just in a dish. That's why we collaborate also closely with other groups in Nijmegen and outside that work on mice models and flies. And then really the last slide, I think that's also just showing where we are going. And it is an example of a project that we are setting up with some collaborators, where we're trying to set up a research a group consortium where we try to really integrate all the different aspects of research, where we, from the same individual, we can now take stem cells, analyze it at different levels, but also at the same time use measurements like EEGs, and also correlate this to clinical assessment. And what we think is that we really need all these different levels of assessment to ultimately come to certain choice of treatment that will then can go back to personalized treatment option. This, of course, something that is ongoing, that will take still many years, but I think this is just to show you the direction that we all want to go to. Of course, I don't do this alone. This is, of course, the new way we are making group pictures those days. But of course, a lot of the work that I've shown you today is done by very young, very enthusiastic people. Well, I think also the collaborators in Nijmegen especially, and the people that have supported this work financially. And thank you for your attention.
Great. Thank you so much. And many thanks to Dr. Buttermore and Dr. Kasri for those talks. I'll direct you to two questions in the chat, one about specifically which specific variants, EHMT1 variants, were studied, and also another question about utility of stem cells. Is there any utility in siblings, unaffected siblings, who have banked cord blood cells, for example, in using them for therapeutic or research purposes? So there are two questions I'll direct in the Q&A for both of you. Okay. And in the interest of time, we'll move on to our next speakers.